again, the main 2 points are that you want you're saying the nuclear membrane goes away, and the chromatin gets packed into chromosomes. And why do you think the chromatids are identical? Go ahead and think about it for a second, and I'll let you think about it, and then I will let you share with a partner. So think about it. Why are chromosomes identical?
Chromosomes?
Or why are these chromatids here identical? What purpose do you think they would serve to be identical? For the future of mitosis. Okay, so talk with your partner. Okay, go ahead and talk with them. I'll let Nick share. Why are these chromatids identical?
They're the same DNA.
They're the same DNA, and why would they need to be the same DNA?
And that they can split.
Yeah, so they can split. AG, did you have anything else?
So when they're pulled apart in mitosis, it's the same on both sides?
Yeah, that's it. That's a good general explanation. So that when they're pulled apart, they're the exact same on both sides. And now we're going to get to a spot where we explain how it separates into both sides and becomes exactly the same. So we can say, so they can split. Into identical cells. I guess, so that they can split and be a part of identical cells, I should say. All right. Next comes metaphase. This begins to be where you can see how it might become it might form 2 identical cells. Okay. Meta actually means after. So you have before, then after, right? So before and after they have come together, and now, what do you notice about the arrangement of these chromosomes, Turner? Any other ideas? Brian?
They're in order.
They're in order. What kind of order are they in?
Red, blue, blue, red.
American.
Yeah, red, white, and yellow.
Okay, small to big. Actually,
James big.
they don't necessarily have to be small to big. They could be, they could be big one, small one, big one. Oops. That was a horrible one. Big one, small one. It doesn't necessarily matter what order they're in. Any other thoughts? Missing the main thought here. Okay. Yeah, they're sideways. And that's going to be important for, we're going to look at another process that, okay, the main purpose of, does anybody know the main purpose of mitosis? To make new cells, and how are the cells supposed to be? Identical, the same. There's going to be another process whose purpose is to actually get as different as possible cells. They're going to so, what else is the same? What else do you notice about this?
Symmetrical.
Symmetrical.
There's 2 blues and 2 reds on the
Blues,
outside.
2 reds, sure. And technically, the color is just so that we can see that they're different. Maybe it's too obvious for you guys to say. I mean, I haven't heard it yet.
They're both the same, or they're all the same size.
So they can do
Another
Like
consistency.
Joey.
They're all lined up.
They're all lined up where?
In the middle.
In the center, the
Yes.
middle.
And
They,
they're dancing.
yes. They straddle the blank of the cell.
Straddle the outside, inside, middle.
Spindle.
Middle,
center.
center. I'm gonna give you. It's actually says equator.
Yes.
I said that.
Oh, Catherine's gravy.
I like gravy.
Why would we need gravy?
I don't know.
Sorry, we're
but
just kidding.
Catherine
Jeez.
wants some gravy. All right. So the chromosomes straddle the equator. Shh, quiet. Chromosomes straddle the equator. Am I on?
I can
Okay,
do
good.
that.
Chromosomes straddle the equator. This is due to the adding or taking away of
Proteins.
protein.
Yes.
Good job, Derek. Good job.
I knew it.
Okay, so they, at some point, here's how it works. You saw it before. This, they are not lined up, right? So these spindles, they'll eventually hook up, and some of these spindles, like maybe this one, if this needs to go over, let's say maybe this one right here. If this needs to go up a little bit, a spindle is going to attach down here and pull up by taking some away, and this centromere down here might add some. The one down here might add some fibers or some proteins to the microtubule to make it longer. So that's how it arranges. You know how it's kind of like a fireman ladder. You know those things they can either lengthen out or shorten, that sort of thing. So that's how they get in the middle like this. All right. Next, another thing. Meta. You could also think of in metaphase. They're in the middle. Okay. Middle. That's a good way to remember. Now, next is anaphase. Anybody know what ana means?
The split.
Kind of.
Split at the beginning.
Kind of.
Different.
What's that?
Different.
Different. You're getting there.
Means the sister
Apart.
chromate.
Apart or away, or anti. So, ana means like anarchy, to go against the government. And I don't know. Anastasia. I don't know how you put that in there. But okay, yeah. Anaphase. So in that phase, in anaphase, they're backing up apart from the equator here. So they were, stop talking.
Derek.
No, just some people over there.
Yeah.
Surprise.
Try to focus.
Okay. All right. So there was an equator along here. They were lined up along there, but now those microtubules, the spindles, have pulled them apart. So it's kind of crazy. They're starting to move away. What do you notice about the types of chromosomes we have here along these spots?
They're in deaf shapes.
How does this one compare to this one? They're the same. How does this one compare to this one?
It's the same.
It's the same thing.
No, it's not.
So
Yeah, boy.
yes, it is.
It's called a flaw.
What?
The bottom one's pointing that way. The top one's pointing down.
Well, it doesn't matter how they're arranged. They're actually, guys,
That's called a flaw.
they're actually bent. That's a good observation. They're bent this way because have you ever seen those movies where somebody gets, they're performing on stage and they're horrible, and then on the cartoon they have the cane that yanks them off stage,
Yep.
and their body kind of does one of these things, right? It's kind of the same thing
Break 'em in half.
with these. Because they're getting dragged by these microtubules away, this end has a little bit of inertia, and this one is getting pulled toward it, so it naturally bends like that. It doesn't
Bending
affect the
down.
performance or the efficiency of the DNA. It's just getting pulled over, okay? So that's anaphase. Centromeres. But what was holding them together before? These sister chromatids? The centromere was. Well, The centromere needs to break, right?
So it splits at the
If,
beginning.
if these things are going to split, the centromere needs to break. So the centromere splits at the beginning,
Chromatids.
then the sister chromatids separate and move toward opposite,
And
sure, poles. I have the opposite poles where the centrioles are. Now I'm not talking about like a flagpole. I'm talking about an
you mean the
opposite.
North to South Pole?
Yes,
Santa
North and
Claus
South pole.
and Miss
There are
Claus.
no actual like
Santa Claus piece.
lengthy poles at the North Pole. It's a pole means opposites, opposite ends. Okay. So the chromatids go toward their opposite end. Uh-oh. Chromatids go toward their opposite end, toward their respective pole. Now, there is one set of single-stranded blank at each end of the cell. Chromosomes. And this, do you remember yesterday when I talked about how the definition of chromosome is a little bit wishy-washy?
Which was
Yes.
no.
It's because chromosome is kind of defined as a set of DNA where it's a full set of DNA for that chromosome. So the sister chromatids, now that they're not together, now that they're kind of separated, they're titled. Their nature didn't change, but their title has now changed from just a chromatid of the same DNA, sister to the other, into, okay, they're on their own now. Now they are the chromosome. Does that kind of make sense? Okay. So now they were called chromatids when they were together, but now they're separate chromosomes because they're separate. Okay? So they are now chromosomes at each end of the cell. Okay. Telophase or telophase. Anybody have any thoughts of what? Give me some words that have telo in them.
Telephone!
Telephone.
Teleporter!
Teleport.
Telegraph.
Telekinesis. Yes, it applies.
Telegraph.
Telegraph. Telepathy.
Telegram.
Telegram.
Cyclops.
Teleperson.
Telescope.
Telescope.
Teleperson.
Teletubbies.
Teletubbies. Television. What in the world does tele mean? What in the world does tele mean? No?
Telephone.
Any thoughts?
Multiply.
Stop. Hey guys, stop talking unless you have ideas. Raise your hand if you do. Ethan.
Translate.
No.
Telephone pole.
That is not a definition of tele.
Move.
No. No.
Wire.
What do all of those things have in common? You might not have put it all together, but each one of those applies. I ran them through my mind when each one of you said those things.
Connection.
No,
No physical connection.
you are getting closer.
Wireless.
No,
Because we just said that.
Multiplying.
tele. Tello.
Visual.
Nope. Okay. Nope. No. Okay.
Separated?
Tello means far away. Television. See something far away. Telegraph. You see you send a graph to someone from far away. Telephone. Phone means hearing. You hear someone from far away. Telescope. Scope means to see. Telescope, you see something far away.
Like a
What about
galaxy.
Teletubbies?
Telephase. The phase where, what's that?
So you can see them far away
Something
too.
separates?
No, this doesn't have anything to do with seeing. Tell
Far
us.
away
Yeah.
phase.
The far away phase. The phase where
It's far away.
the chromosomes are far away from each other. Okay? Tele means far away. Telophase. So we can say chromosomes are far from each other. They're on the far ends of the cell that's about to split. Okay? So telophase, it is when the cell officially begins
Split.
to split. Good. If it's an animal cell, the plasma blank pitches together.
Membrane.
Membrane. But with plant cells, it's a little different because cell walls can't bend. They have those fibrous layers, right? So they have too much support. So cell walls in plants can't bend like that. So a cell plate starts forming in the,
Cytoplasm?
here's a cell plate,
Middle.
in the middle, center, whatever.
Center.
So here's our cell plate that begins to form in the center, and it actually grows out. This, in an animal cell, it pinches in. This one, it starts in the middle and grows out. That's kind of interesting. So these are just a few different views of telophase. So it pinches in. In plant cells, it grows out. Okay? Now, while these cells are splitting, they reverse what happened in prophase. Because what's in between these stages? The in-between phase? Like interphase. The time in between the mitosis stages is when the cell does its job. Okay? So, in order to do its job, what needs to happen to the DNA?
It needs to be destroyed.
No, it can't.
Needs to be replicated.
Needs to be-
At some
blueprint?
point, if it needs to go to mitosis, it will want to.
So all of these steps fall under mitosis?
Yep.
Oh.
It needs to split.
Yep.
So
All
it's
of
like
these
the
are-
one big
separate.
step and these are all like little steps already.
Yep. Yeah.
Needs to separate.
And I guess that's a good point to make. All of these are types of mitosis. Parts. Did I say types? Okay, sorry. All of these are parts of. All of these are parts of mitosis. We'll get there though. While cells split, they reverse what happened in prophase. Well, what happened? What were the two main things that happened in what's one, Kylie? One thing that happened in prophase? The DNA gets packed. So if this is reverse of prophase, what's going to happen then, Naomi? It loosens, sure. It unravels, maybe. It unpacks. So, oops. So chromosomes unpack into, what's it unpacking to, Catherine? The thready stuff, chromosomes.
Spindles.
We said in prophase, something packs up into chromosomes. Yes, chromatin. Chromosomes unpack into chromatin. That was the opposite of prophase, right? Prophase, chromatin packed into chromosomes. Now in this phase, telophase and stuff, it actually unpacks, it unravels so it can be used, right? The other thing that happened in prophase, what's that, Chapo? We said chromatin packed into chromosomes. What's the other thing? The other main thing.
They separate.
Separate. Which, are you looking at prophase? What's the other main thing that I had you guys search?
Membrane of the nucleus goes away.
There you go. In prophase, the nuclear membrane goes away, so Turner, what's going to happen?
Comes back.
It's going to come back. Nuclear membrane forms around chromosomes instead of going away. And then, because the membrane's back, the DNA is unraveled, guess what it can do?
Wind up again.
It could wind up again. But first, it's going to do whatever it's made to do. It's going to do its cell thing. If it's a muscle cell, it's going to do its muscle cell thing. Okay? So let's recap this. What's what's the first phase, Kelly? All right, prophase. And what are the two things that happen in prophase, Kelly?
The membrane disappears and it comes inside the chromosomes.
Nuclear membrane disappears and chromatin packs into chromosomes. Okay. Mitosis. What? No, I'm crazy. Metaphase. Jordan, what happens in what's the main thing that happens in metaphase? Actually, they don't split yet. Metaphase, they go to the middle. So line up in the middle. That's the main thing for metaphase. It's basically a, hey, we're setting this up to be able to split perfectly. The next one, Josie. Anaphase. And what do they do?
They're like separate.
Yep, move apart. We could say chromatids move apart. And become, now titled,
Single-stranded.
single-stranded chromosomes. Yep, you could say that. No, not yet, because they're still packed. Move apart and are called chromosomes again. I won't say it again, but they're called chromosomes. The next one, Joey.
Telophase.
Telophase.
Picture looks like a telephone.
Yeah. So, and what's the main thing that happens in telophase, Kelsey? Yeah. It officially splits. Starts splitting. And the other thing that happens, Aaron? How does it relate to prophase? In a lot of ways. Yep, it unpacks the what? Chromosomes and turns it into protein. And yep, nuclear membrane comes back. So let's say it starts splitting and reverses prophase. So that's kind of a summary of today. Now let's do a couple. Oh, I don't have all the eggs out. If anybody doesn't have an egg, I'm gonna get one.
Jacob, you sit down for grandma. I'm sitting out. Jacob, you Jacob, you don't need no fun.
Learn to tell. Good job.
Jacob.
Take this one.
What if there are no more?
Jacob.
Oh,
This one was over here first, Travis.
try these two. I took most of them out, but don't worry. I mean, I hope the rest work. Okay. Anybody else need one?
Christian, I need one. Turner, you got one?
Turner needs one.
Hang on,
Kelsey,
why did you do
Turner
that?
needs one.
Bag.
Any extras lying around? No? Sorry, Turner.
I know the answer to
Okay.
that.
Everybody voted? Okay? Where in the cell are cells chromosomes when they are ready to split? Not when they have actually split. Okay? So when they're, if they were along the edges, they have already split, right? So we know that when they're ready, they're lined up in the middle like this. And so what phase is that called?
Metaphase.
That's the question.
Metaphase.
The one that we just talked about. What phase was just described?
When they're lined up in the middle.
Yes.
Oh, crap, no.
You can change it.
Okay, let's get that last one in. Okay, all right. Vast majority got it, good. Wait, yeah, metaphase is right. I guess we'll try and do the last one. There was another question, right? Okay, well,